This video will be about how protein application and physiological differences between prokaryotes and eukaryotes determine the molecular components involved in protein expression vectors. You've seen this chain of information flow multiple times. For protein expression vectors, both RNA and protein are essential goals. In summary, with protein expression vectors, you clone a template into the plasmid or vector in such a way that the template can be transcribed to give out an mRNA which has all the necessary information to recruit ribosomes and result in your desired protein. Why would you want to do this? Broadly speaking, there are two reasons. One involves production, manufacturing, and therefore purification of your desired protein. The other is for in vivo use. On the production side, you are likely trying to make an enzyme for research purposes or commercializing a therapeutic one. This protein can be an antibody, a therapeutic protein like insulin, vaccine, or something similar. You also want pure protein if you want to do protein sequencing. Sometimes virus production requires pure proteins too. Something like crystallography or cryo-EM, where you study protein structures, also need pure protein. For in vivo use, you may be looking to express a gene inside a cell as part of a gene therapy treatment. Maybe you're interested in understanding the function of the same protein, and this may involve a reporter system, microscopy, and tracking the desired protein. Alternatively, you may just want to heterologously express a protein. And there are many other applications that fall into this list, but I hope you get some idea. Let's envision the realistic expectation when working with protein expression vectors. The first expectation is around the origin of protein. This could be from a prokaryote or a eukaryote or a viral species. You have the protein origin, but now you want to express this protein in a host. And your options are a prokaryote like E. coli, or eukaryotes like yeast, insect cells, mammalian cells, or perhaps even plant cells. Let's go a step further. You may be interested in expressing the viral protein in either a prokaryote or a eukaryote. You may be looking to express bacterial protein in a bacterial host, which is not that complex, or a eukaryotic protein in a eukaryotic host, which can be complex. You could do cross-species expression as well, which is called heterologous expression. And this is where things get extra challenging. A wildcard situation is to express a protein without a host. This is called cell-free or in vitro translation, which works on a similar principle to in vitro transcription. You can express most any protein with this method, but it's not perfect. By and large, there are only two types of protein expression systems. So let's dive into their physiology. Bacteria have a dual membrane structure. In between the outer and the inner membrane is the periplasmic space. There are no organelles, which means that the ribosomes make proteins co-transcriptionally. And if the protein stays in the cytoplasm, it is a cytosolic protein. This protein, however, could also be secreted. In some cases, a protein can also be periplasmic. It can also bridge both inner and outer membranes. It can be on the outside of the outer membrane. It could be on the periplasmic side of the outer membrane. It could be on the periplasmic side of the inner membrane, or on the cytosolic side of the inner membrane. So many options. Bacteria also have a lot of lipopolysaccharides on the outside. So generally, a lot of proteins on the exterior side carry interesting modifications. Now, eukaryotes are different. We have got complex physiological compartmentalization. The RNA is made in the nucleus, and it is transported out into the cytoplasm for translation. So, of course, like bacteria, we have cytosolic proteins. Quite a lot of proteins are made at the endoplasmic reticulum. These proteins could end up in vesicles, mitochondria, or even the membranes, so they can be secreted out of the cell. The membrane proteins can span the membrane, be towards the cytosol or towards the exterior. Some proteins can be sent back into the nucleus, like most polymerases, or any other organelle. Essentially, we're saying that the amount of protein trafficking is huge in eukaryotes, given the membrane-based compartmentalization, whereas bacteria do not have this complexity. When it comes to protein expression, our concern in eukaryotes is with protein trafficking, the size of the proteins and their complexity. Typically, over 100 kilodaltons is said to be a big problem. Eukaryotic proteins also tend to have a lot more disulfide bonds, high meaning more than 2 or 3. Eukaryotes, especially mammals, have very complex post-translational modifications, and glycosylation is the most common modification, and this typically becomes a problem in other organisms. The amount of proteolytic processing is also quite high in eukaryotes. Essentially, all these plus points of eukaryotes are a limitation for the bacterial system of protein expression. It cannot handle proteins more than 100 kilodaltons in size because it runs into the issue of improper protein folding. Bacterial proteins do not have a lot of disulfide bonds. The only place for making such bonds in bacteria is the periplasmic space. This is because the cytoplasm is reducing and you don't have any organelles to compartmentalize. So the only space left is the periplasm, which is oxidative in nature. Here you find a lot of electron acceptors that can take two cysteines and join them. In bacteria, the type of glycosylation is different and sometimes just not even possible. Bacteria have a lot of lipopolysaccharides on them, and it sheds these molecules. Any contamination from lipopolysaccharides in your therapeutic protein production can cause an immune reaction. These differences are not just between prokaryotes and eukaryotes. You can have differences within eukaryotes. The level and complexity of glycosylation is widely different from yeast to mammals. If you use insect cells for protein expression, you grow them at 27 degrees Celsius versus 37 for yeast and mammals. So differences come from that too. All these weird modifications, if not corrected, can lead to toxic reactions for heterologously expressed therapeutic proteins. The low growth temperature of insect cells can actually be a good thing if you're trying to make complex proteins with complicated folding behaviors. Plants are not super commonly used, but they do have their use cases. In general, you get very low yield from them. Outside of this, cell-free translation works too. It is quite expensive to do and has poor yield, but if you want to do something quick, it can be useful. Let's talk about the design of the protein expression vector. First, you need the template DNA that makes the protein. As we shall see, for protein expression vectors, we work our way backwards to get the template. Once you have the template, you have some idea about its size and complexity, which also reflects the RNA size and complexity. For templates in protein expression vectors, a big jump comes from understanding the gene structure, particularly at its RNA level and especially for intron-containing genes. You also have other components that you have to worry about as well. When doing heterologous expression, codon bias and its optimization becomes a challenge. On the vector side of the things, you need to understand its size, complexity, stability, and its copy number. Typically, protein expression vectors are kept at around 100 copies. And just like we saw in RNA expression vectors, you need the rest of the appropriate elements in your vector. The difference between RNA and protein expression vectors comes from the insane amount of extra engineering options you have to think about. This primarily can be categorized based on the use of your protein. Depending on either production purification or in vivo expression, the extra engineering will change. For purification of a specific protein from the cell, your job is to avoid the rest of the proteins in the cell. How do you do that? The most common way is to add an epitope or a purification tag that can be selected for. Sometimes you're interested in secreting the protein, and maybe normally your protein of interest doesn't get secreted. In that case, you have to engineer a secretion tag. If you want to change the localization of a protein, say you're heterologously expressing a protein with disulfide bonds in bacteria. In that case, you have to shunt the protein to periplasmic space so it can get oxidized and form disulfide bonds. Other times, especially for in vivo expression, you may have to create fusion proteins. In that case, you may need linkers, bicistronic systems, or cleaving domains. If you want to control degradation of a protein, induce it at a specific setting, change localization, add an epitope for staining in Western blots, or microscopy, any modification to the protein will require extra engineering. In the next video, I will discuss the molecular components in much more detail.